everyone. Another global immunotalk. Wherever you are joining, we are glad that you are participating today. Okay, so just a reminder that the next Global Immunotalk will be by Scott Mueller and is part of the Global Live initiative. So that talk will be will go live at 4 p.m. Australian Central Time, and it will be available again at the regular time that you are joining right now, so 9 a.m. PST, noon EST. Okay, so without further ado, I would like to introduce to you our fabulous speaker today, Dr. Maria Rescigno. So Maria is originally from Italy, Naples, and she has graduated in biology in 1990 at the University of Milan. From 1991 to 1994, she worked at the University of Cambridge in UK in the Department of Biochemistry as a visiting scholar. And afterwards, she worked at the National Research Council of Milan, where she received her PhD in pharmacology and toxicology in 1999. From 1999 to 2001, she worked at the University of Milano Bicocca, where she specialized in applied biotechnology. From 2001 to 2017, she was the director of the Dendritic Cell Biology and Immunotherapy Unit at the Department of Experimental Oncology at the European Institute of Oncology. From 2018 till present, she became full professor and group leader of mucosal immunology and microbiota at Humanitas University in Milan, where she is also since 2019 the vice rector and delegate for research at the same university. Maria's lab focuses on mucosal immunology, and in particular, the interaction between host and microbes at mucosal sites. She was the first to show that dendritic cells actively participate to bacterial uptake in the gut, and that the local microenvironment dictates immune homeostasis. She also discovered the existence in the intestine of vascular barrier that resembles the blood-brain barrier and that restrains bacteria from entering the bloodstream and disseminate systemically. Another field of interest for Maria is the development of new cancer immunotherapy strategies based on bacteria as immune stimulators. She has shown that bacteria can drive the establishment of gap junctions between tumor cells and immune cells for an efficient priming of anti-tumor immunity. She also developed tumor-specific bacteria that act as intelligent missiles to kill only tumor cells. So Maria communicated this terrific research in more than 130 publications, including many papers in top-tier journals. She has also been awarded numerous prizes, including, just as an example, in 2006, being elected of EMBO as EMBO Young Investigator, and a few years later, in 2011, she was elected as a member of EMBO. Maria is also, since 2019, member of the advisory board of L'Oréal UNESCO Prize for Women in Science, and she also is a female entrepreneur because she founded and is president of Postbiotica, a spin-off company that is a spin-off of the University of Milan. And she also received a number of awards as part of this company, including in 2017, the winner of My Star in Barcelona, Spain. So Maria, we cannot wait learn from you about the gut axis and microbiota. So thank you so much for your efforts to present at this forum.
Thank you, Elena. Thank you very much for everything and for inviting me here.
It's a pleasure. And Maria, before we move into the talk itself, we always want to learn from our speakers and their experience. So we have a question for you that is, what is the single most important advice you would give to a young scientist starting a career in immunology?
Yeah, the only thing that I want to say is don't give up. Because I think that especially working in immunology is very tough because it's complex. It's a very complex system, and what we do is primarily in vivo, and it takes time. So I have many people, many PhD students working with me, and sometimes they are a little bit frustrated by the research. I just say, don't give up. Sooner or later, you will have your results, and they will give you a lot of pleasure. That's what I can say to the young investigators.
I love it, Maria. That's true. And no, sometimes we forget that, how complex immunology can be, and for an, a student in the first year, and that, yeah, that all gone through this challenge of getting familiarized with the topic. And yeah, thank you so much. I think it's very helpful to hear that. Okay, so without further ado, then I invite you to share your screen.
Okay. I hope you can see it.
Excellent. Perfect, Maria. Thank you.
Okay, perfect. So thank you, Elena, for everything. I will be talking about the microbiota and host interactions in the gut-liver-brain axis. And I wanted to start just by reminding you that the intestinal microbiota, which is the most abundant in our body, has been shown to participate to a series of functions of our body, including food breakdown, which is the most commonly known, but also the immune system maturation, several other functions, and the brain development and function. So I will be talking primarily about these two parts in the context of cancer and in the context of brain behavior. So, as you know, the microbiota is sitting in the gut, and but not everywhere. So there are areas in which there are very few microbes, while there are areas like the small intestine, and particularly the last part of the small intestine and the colon, which are particularly colonized by the microbiota. And our body also has some defenses against the microbiota, and in particular, one of the most important defenses is the mucus, because you can see here, this is the intestinal wall. Here you can see this is the mucus layer is organized in two layers. One inner layer, which is almost sterile. You can see the bacteria are in green. This is a 16S fish analysis. You can see the bacteria are sitting in the outer mucus layer, and they are anchored there, and so they are not eliminated via the peristaltic movements of the gut. But there is an area which is completely sterile, and very few bacteria are capable of crossing this area, and those that do are either pathobionts, so are part of the bacteria that are more pathologic, or can be pathogens themselves. So, but as I said, there are some bacteria actually that can cross the mucus layers, but then they find another barrier here, which is represented by epithelial cells. So this is a monolayer of epithelial cells, which are sealed one to the other by tight junctions. So whatever can cross these epithelial cells has to do in the paracellular route, not the transcellular, so between cells, but across epithelial cells. And then this bacteria can be found within the lamina propria, which is just below the epithelium. But if you analyze at what happens if bacteria can cross the mucus and the epithelium, they mostly get into the mesenteric lymph nodes. But if you look at the anatomy of the gut, You can see here that these are in blue are the epithelial cells, in white are the blood vessels, and in red are the lymphatics. So to get in mesenteric lymph nodes, bacteria have to go through the epithelium, but also they have to pass through the blood endothelial cells and then get into the mesenteric lymph nodes via the lymphatics. So how come that very few bacteria get into the liver and most of the bacteria get into the mesenteric lymph nodes if the blood vessels are close to the epithelium and the blood vessels through the portal circulation can reach directly the liver? But you can see very few bacteria that are getting into the liver. Primarily pathogenic bacteria like Salmonella typhimurium, for instance, are capable of doing that, but not very few bacteria from the commensal flora can get into the liver. So we described now in some years ago, in 2015, that just below the epithelium, we have an additional vascular barrier that we call the gut vascular barrier. And this barrier actually resembles a little bit the blood-brain barrier. The reason being that it has endothelial cells, as expected, by the blood vessels, which are surrounded by pericytes, and they also have enphytes from enteric glial cells, which are the equivalent of the astrocytes. In addition, between endothelial cells, we have tight junctions that don't allow the passage of molecules between cells. So whatever enters has to go through the endothelium. And still in analogy with the blood-brain barrier, we know that in the blood-brain barrier, when it is established, there is a change of the beta-catenin Wnt signaling pathway. So when it is off, the barrier is not formed yet. But when the beta-catenin pathway is activated, then the barrier is organized. And this correlates with the reduction of a marker, which is called the PV1. So if PV1 is high, the barrier is compromised. If the PV1 is low, the barrier is established. So similarly, we found that in the intestine, the duodenum, jejunum, and ileum, we couldn't see these markers. So suggesting that since we don't see the marker, that this barrier is established and that beta-catenin is on. And so the barrier does not allow things that are accidentally or purposely cross the epithelium to get into the blood circulation and then disseminate systemically. So what is PV1? So PV1 is a molecule which makes the diaphragms of the fenestre. So it actually makes a sort of a sieve that controls what enters the endothelial cells via this fenestre. But when we analyze the expression of PV1, actually, we do analyze the expression that is when this antibody, which is called MECA32, which recognizes a PV1, but only when the PV1 is in open conformation. So meaning that there is not this sieve formation anymore, but the PV1 is accessible to the antibody. And so we can see that the PV1 is in open conformation. It is like opening these holes and allowing things to get through, and there is no sieve any longer. So what we did was we treated animals with Salmonella, and then at different time points, we analyzed for the capacity of this Salmonella, since we know that it can disseminate systemically, to have an effect on the vascular barrier. And what we found that very quickly, already at 2 hours, we start seeing the detection of PV1, so meaning that the barrier is becoming compromised. And this is very clear at 6 hours. So when we have Salmonella infection, Salmonella can cross the mucus, and this was known. It can cross also the epithelium, and it can also modify the gut vascular barrier. So it opens the vascular barrier, and this correlates with the capacity of Salmonella of inducing the release of transaminases, which are liver enzymes that when you find them in the blood, this means that the liver is damaged. So already at 2 hours, so when we start seeing this modification of the barrier, we start seeing already some release of transaminases. And this correlates with the capacity of Salmonella to get into the liver, mostly already at 6 hours, but mostly at 24 hours, and then in the spleen later on. So suggesting that when you have salmonella infection. This changes the vascular barrier, and through the portal circulation can get into the liver and then eventually also disseminate to other organs, including the spleen. So why am I telling you this? I'm telling you this because this vascular barrier may have several functions, and when it is disrupted, may be involved in some disorders or some phenomena. So we decided to concentrate on two major topics, which was, is this gut vascular barrier involved in the metastatization of colorectal cancer from the gut into the liver? And the second is whether it can also allow an interaction between the gut, liver, and the brain, and whether this is, can be modulated by the opening or closure of the gut vascular barrier. So regarding the first task, as I said, we know that this barrier is present in the gut. And since we know that when you have colorectal cancer can actually metastasize to, through two major routes. One is via the lymphatics into the mesenteric lymph nodes. And we hypothesize that in order to get into the liver, this dissemination goes through the blood vessels, and the blood vessels have to be compromised, because otherwise we would have the gut vascular barrier, which would block the capacity of tumor cells unless they can modify either themselves or indirectly and indirectly the vascular vessels and then get into the liver. And if this is true, we were expecting to see also a change in the vascular permeability. So when you analyze patients with colorectal cancer, these patients are stratified according to whether they have lymph node metastasis or distant metastasis, or whether they do not have any lymph node or distant metastasis. And this decides what type of treatment they will have. In one case, if they do have lymph node metastasis, then they undergo chemotherapy plus targeted therapy. If they don't have lymph node metastasis or distant metastasis, they either have no treatment or, according to the oncologist, they may have only chemotherapy. But unfortunately, nearly 50% of patients that don't have distant metastasis at the time of primary tumor resection, they will develop distant metastasis within 5 years of follow-up. So suggesting that at the time of resection, they were already metastasizing, but their metastases were not visible yet via the common ways of detection. Because while the lymph node, it's easy to be detected because you can take the draining lymph nodes, the sentinel lymph nodes, and just dissect it. If you have metastases in the liver or in the lung, these are very difficult to see if they are not large enough to be seen via imaging techniques. So we hypothesized the following. So going back to the vascular barrier and to the capacity of bacteria to modulate this barrier, that during tumor development, primary tumor development, you also have an intratumoral microenvironment by the microbiota, because we know that the microbiota can also enter the primary tumor. And this microbiota may modify the vascular barrier and migrate into the liver. And then this is the first event for tumor dissemination, because then you create within the liver an environment which is then driving the recruitment of tumor cells within the liver. So this was our hypothesis. So in order to analyze whether this hypothesis was correct, we used two model systems, so the human system and mouse. For the mouse, we know that since we know that the microbiota can be modified in colorectal cancer patients, and this is due to the fact that the mucus is compromised and the microbiota can actually penetrate the epithelial layer and can be found within the tumor, that this may have an involvement. So we used a model of colorectal cancer, which we generated in the laboratory. And in this model system, we crossed APCM in mice that are normally, they have, in blue, they have tumors primarily in the small intestine and very few tumors in the colon. We cross them with the C3a receptor knockout, which gives a change in a tumor location. So from the small intestine, the tumors are found primarily in the colon. You can see here. And so this is a perfect model system to study what happens in the gut and eventually also in the liver. So looking at the gut, here is the analysis of the expression of PV1. I remind you that if PV1 is visible, this means that the gut vascular barrier is compromised. So you can see here that in the mice that develop tumors, even in the absence of any metastasis in the liver, you can see that you already see a change in PV1 and the detection of PV1, which is not seen in the other mice. We couldn't see in the APC-min mice because the APC-min mice unfortunately die before APC-min C3A receptor knockout develop tumors, so they cannot be used as a model system. However, when you look then in the liver, we found that the capacity of the presence of the tumors within the gut correlates with an increased number of bacteria within the liver. So suggesting that when you have tumors, primary tumors, you also have some bacteria that are migrating from the gut into the liver. And you can see here that this is the case of the mice that develop tumors in the colon. And this also correlates with an increase of recruitment of inflammatory cells, including macrophages, neutrophils, and inflammatory monocytes, specifically in the mice that develop tumors in the colon. I remind you that at this time point, these mice don't have any metastasis in the liver, so this event is occurring before any tumor metastasization in the liver. Unfortunately, these mice do not live enough for having metastasis in the liver, so we find only very few mice, and this is not sufficient for us to make any statistical analysis. So we switched to another model to see the role of the microbiota, the intratumoral microbiota in liver metastasis. And in this case, we use mice in which we generated tumors, primary tumors in the gut via AUM-DSS, which is a classical model for generating inflammatory cancer-derived inflammation tumors. And then we injected these mice within the liver with luciferase stained tumors, and from the in the spleen, and from the spleen they get into the liver. And we asked ourselves whether this was dependent on the microbiota. So first of all, we found that if you have tumors within the gut, so if you have primary tumors in the gut, you also have many more metastases in the liver. So suggesting that the presence of tumors in the gut also favors the development of liver metastases from the spleen into the liver. And this is not just dependent on the inflammatory reaction, which is induced by DSS, because you can see here that this is very similar to the untreated mice. So they have a very similar amount of liver metastases. So the metastases are formed, but they are formed many more if you have primary tumors in the colon. And what happens if we give antibiotics? If we do give antibiotics, we drastically reduce the number of liver metastases. So suggesting that when you have bacteria in the gut, or we give antibiotics which can go anywhere, so bacteria can actually be eliminated everywhere, then you also reduce the number of liver metastases. And indeed, we also have much fewer numbers of liver bacteria in mice treated with antibiotics, and this also correlates with a reduction of macrophages, neutrophils, and inflammatory monocytes in the presence of the antibiotics. So then we said, well, this is a mouse model, what happens in patients. And so we did the retrospective study of colorectal cancer patients, and we analyzed the expression of PV1 on paraffin-embedded tissues samples from 180 colorectal cancer patients that will develop or not distant metastasis in the follow-up time, but they didn't have any distant metastasis, only lymph node metastasis at the time of resection. So by the time of resection, we couldn't say whether these patients would have metastasized or not. So we analyzed the detection of PD-1, and you can see here that those that will develop distant metastasis, they have much more expression of PD-1, which is not related to the number of vessels depicted by CD31. So you can see that patients that will develop distant metastasis already in the primary tumor, they had a high expression of PD-1. And if we stratify the patients now according to the detection of PD-1 and the presence of lymph node metastasis, you can see that patients that had no PD-1 or low PD-1 expression and no lymph node metastasis are the one that in the follow-up time of 10 years, they do best because they have reduced chances of having either lymph node metastasis or distant metastasis. By contrast, those that have either lymph node involvement or high PD-1, but not the other parameter, they are doing a bit worse than this. But they are also, we did also a multi-parameter analysis, and these are independent factors. But if you look at patients that have a high PD-1 and also lymph node involvement, so they have the two ways of metastatization at the same time, so via the blood vessels and via the lymphatics, these are the patients that do worse. And we couldn't say this if we didn't have the marker for an increased endothelial permeability. And this also correlates with an increase of bacteria within the metastatic liver lesion as compared to the paired healthy liver parts. You can see that in the tumor foci, you can see many more bacteria there. So indicating that what we found in the mouse system has a counterpart also in the human. So this is the summary of this first part on the involvement of gut vascular barrier in the metastatic process from colorectal cancer. So if you have a colorectal cancer, the first event is the modification of the gut vascular barrier, and this is characterized by the dissemination of bacteria within the liver, even before tumor formation. This dissemination of bacteria in the liver is associated with a recruitment of inflammatory cells and the formation of premetastatic niche. And this premetastatic niche then drives the recruitment of cancer cell from the primary tumor into the liver for the formation of metastasis. So the gut vascular barrier is involved in this process because it is modified most likely by the microbes that are migrating into the liver. Here they are recruiting inflammatory cells. They are inducing the formation of this premetastatic niche, and this is then recruiting tumor cells from the primary tumor into the liver. So I hope I have convinced you that this is an event that is occurring during metastatic formation of colorectal cancer. So the second part I wanted to talk to you, it's still on the gut vascular barrier, and it's going up from the liver into the brain. Not talking now regarding the metastasis formation, but I wanted to stress more on another disease, which is the inflammatory bowel disease. We know that in several disorders of the inflammatory bowel disease and also other disorders, there is what is called the leaky gut. So you have an increased permeability of the gut. And during inflammatory bowel disease that we know that is divided into Crohn's disease and ulcerative colitis, many patients have liver disorders, so they can have gallstones, they have PSC, primary sclerosing cholangitis. They can, in ulcerative colitis, you can also have portal vein thrombosis, liver abscesses. So many patients that have inflammatory bowel disease, they also are characterized by liver disease. But another extraintestinal manifestation of this disorder seems to be brain issues. So in particular, neuropathy, myelopathy, stroke, and many of these patients with inflammatory bowel disease have several problems, neurological problems. And in addition, 34% of patients with active IBD present with depression. Depression has always been seen as one of the price that patients pay because of the presence of the disease, because it's a debilitating disorder. And so probably the depression and the anxiety are associated with the difficulty of the patients to cope with this disease. So we analyzed what is the GVB status of these patients. And we analyzed healthy controls and ulcerative colitis patients, both the ulcerated areas and the distal areas. And you can see that PV1, so when PV1 is high, I remind you that the vascular barrier is compromised. You can see that both in the ulcerated areas and the distal area, the PV1 is high, so suggesting that the vascular barrier is compromised also in ulcerative colitis patients. And then we use the mouse model system. We use the DSS mouse model. In this DSS, mice drink in the drinking water, you give DSS, which creates holes within the mucus, and this allows bacteria to cross the mucus and so to initiate an inflammatory response within the gut. So you give it in the drinking water for 3 days, and then we give water for another couple of days, and we analyze what happens in these mice in the gut vascular barrier. And you can see here, this is the control mice, so at time 0, so when they have not yet received the DSS. And you can see that the PV1 is undetectable because the gut vascular barrier is in place, is normal. But if you already, 1 day after DSS colitis, you can see that you can see an increased detection of PV1 here in magenta. And this is also at day 3. But then when we give water, we still see that the PV1 is high. So the gut vascular barrier is not yet recovered after 2 days of water. By contrast, what we see is that in the epithelium, we can start seeing the expression of tight junction proteins like ZO1, so suggesting that the epithelium is the first to recover after in the recovery phase, so when DSS is removed, and so when you have water, while the gut vascular barrier is not yet modified. And this is just the quantification on several sections and mice. And this also correlates with an increase of IL-6, which is an inflammatory cytokine. So when you have inflammation, we also have an increase of IL-6, so suggesting that the presence of inflammation in the gut correlates with a systemic response, inflammatory response. So then we asked ourselves what happens in the tissues. So we analyzed in the colon the presence of inflammatory innate cells like monocytes, macrophages, and neutrophils. And as expected, when you have induction of colitis, you also have an increase of inflammatory cells, including monocytes, macrophages, and neutrophils. But then we analyzed in the liver, since we had this systemic inflammatory response that was ongoing, as suggested by IL-6. And you can see here that in the liver, we also see the recruitment of inflammatory cells. But then unexpectedly, we found that also in the brain, we could see a recruitment of inflammatory cells. So suggesting that when you have inflammation ongoing in the intestine, this can spread across the gut, liver, and brain axis. So then we said, okay, if we have an increased inflammation in the liver, in the liver and the brain, maybe in the brain we do see an increase of permeability of the brain due to the inflammation. And so we performed the following experiment. So again, we have the same treatment schedule. So DSS is given at day 0, and then at day 1, day 3, and day 5. So they from 0 to 3, they have been treated with DSS, and then 2 days with water. And then in different mice groups, and this is important to tell you because at day 0, day 1, day 3, and day 5, different mice groups were injected IV with a dye, 70 kilodalton dextran dye, to see how much of this dye was penetrating in the brain. And so you can see here that at T0, we already could see some penetration of this dye in the brain, which to us was a little bit surprising because the blood-brain barrier has a size exclusion of 500 Dalton, not 70 kilodalton, so much smaller. So to us, this permeability was a bit surprising. We are not experts of brain permeability. Then at T1, we could see still some permeability a little bit higher than at T0, and then at T3 a reduction, a drastic reduction. You can see this blue. You don't see the blue anymore. You can see it only in a particular area. And then at T5, the situation after 2 days of water was restored. And this is just the quantification. So the first thing we analyzed was the blood-brain barrier to see whether there was a change in the blood-brain barrier. And we couldn't really see much changes at these time points. So PV1 was not modified at all. We could see some changes, but later on they were not consistent with this increased permeability that we observed, and a complete reduction of permeability at day 3. And so we went more deeply into understanding where this dye was extravasating. And we found that the dye was extravasating in a particular region of the brain, which is the choroid plexus. So very close to the choroid plexus in these lateral areas, we could see this 70 kilodalton dye that was extravasating. So I'm not an expert at all of the brain. I've been working all my life in the gut, so I had to ask advice from neurobiologists, and so we were helped by Simona Lodato and Michela Matteoli in our institute, and we analyzed what is really the choroid plexus. So if you look at the structure of the choroid plexus, it looks so similar to the structure of the intestinal villi. So you can see here, this is the epithelium, and then the endothelium, which is sitting just below. If you look at the choroid plexus, you can see that you have the endothelium and then the epithelium. Okay, this has always been considered as a barrier, but only for the epithelium, because the epithelium is the one that is filtrating the blood that comes from the periphery and is generating the cerebrospinal fluid, which is then permeating this meningeal area and the ventricles here. But the difference is that in the gut, this is the external world, and so you have first the epithelium and then the endothelium, while in the brain is the blood is the internal world, which for the brain is the external world, but still it's the systemic circulation. And then the epithelium is the second cell type that is found after the endothelium. So here you can find first the epithelium and then the endothelium. Here it's the opposite. And in fact, you can see here, this is the endothelial vessels, and these are the epithelial vessels. If you look at the choroid plexus, you can see that the epithelium is all around. In gray, you can see this is ZO1, which seals the epithelial cells one to the other. And here you can find the blood vessels, which are these ones here. You can see these are the endothelial cells. And we could also look at PV1. So this was the first difference between the gut and the brain, is that PV1 is detectable on these vessels. And this is consistent with the fact that you have to filtrate the blood, and so the vessels have to be permeable. And since you have high PV1, this means that the vessels are permeable. Okay, so then we analyze what happens during the SS colitis and the dye distribution. So again, this is still the same type of schedule. The mice received the SS for 3 days and then water. So this is day 0. This is the choroid plexus, and this is a zooming of this. You can see in magenta now it's the dye. You can see that this is extravasating. And then at T1, you can see that the dye is extravasating even more, suggesting that during inflammation, you have even an increase of dye, which is extravasating, as if this barrier is even more compromised. But then if you look at T3, we were really surprised to see that at T3, when we still have DSS and we are switching to water, but we are still under inflammation in the gut, that you don't see any longer this extravasation of the dye. This is completely clear. And then at T5, you can see that you start seeing again the dye which is extravasating. So there is a time point in which the choroid plexus becomes more permeable, and there is a time point in which the choroid plexus is completely impermeable. So nothing goes through the endothelial vessels. And this is also correlating with the capacity of the dye and to get into the cerebrospinal fluid, because if you consider that then this choroid plexus is filtrating the blood, you're expecting that if you have a dye in the blood, this is also getting into the CSF. And indeed, at day 0, you can find it in the CSF. Then at T1 you see more, and this is also in agreement with the fact that we see more extravasation of the dye within the plexus. And then at day 3 we see no dye or very little dye, and then at T5 this goes back into the CSF. So to make things a little bit more clear, because I understand it, if you're not in the field as I was, it's very difficult to understand. So these are the early time points. So the dye is getting from the blood, because we give it IV, through the vessels, through the endothelial cells and the epithelial cells into the cerebrospinal fluid. And so this is what happens already at time 0. So you have some of the dye that gets into the ventricles. When you have the initial inflammation, there is even more increased permeability, and so this is getting even more into the cerebrospinal fluid. By contrast, at day 3, when during inflammation, these vessels are not allowing anymore the dye to go through, and the dye remains in the vessels. And then, since we are performing a perfusion of the mice, we also don't see the dye any longer. So this is why we see it clear. And so this is actually blocking the dye to get into the brain. And the dye for us is associated to inflammation. So we can expect that in this case, we have an initial inflammatory response, which may explain why we see an increase of inflammatory cells within the brain. But at the same time, we also are trying to block the release of inflammation from the blood into the brain. And so we are trying to protect the brain from further damage, which would arise after the inflammatory response. So since I told you that PV1 is a marker of barrier permeability, we asked ourselves whether these barrier modifications of the barrier were associated to the endothelial cells indeed, rather than to the epithelial cells. And this is exactly the case. As I told you, these vessels, differently from the vessels in the gut, are already permeable at day 0, and this is why we see the dye permeating into the cerebrospinal fluid. At T1, we see an increase of PV1, so as if this becomes even more permeable. And then at day 3, you see that PV1 is completely shut off, so suggesting that the PV1 is, the vessels are closing down, and this does not allow the dye to extravasate any longer. And then at day 5, in which we have, after 2 days of water, they are recovering, and so this is also recovering. We also analyzed which were the cells that were first responding to the inflammatory response, and the cells that are first responding are actually endothelial cells and pericytes, so capillary vessels and pericytes, which are the vascular component is the one that is modified between T0 and T1, and also T1 and T3. And so these are the cells that are first responding to inflammation are actually the endothelial cells, and which are the ones that are more exposed to the inflammatory response. And we also found that the Wnt signaling pathway, which if you remember in the gut vascular barrier, but also in the blood-brain barrier, was important for the regulation of P1 expression, is also modulated, and is modulated. T1 is downregulated, and then the T3 is upregulated again. And this is occurring primarily in the vascular capillary cells. So since we generated a mouse in which we can modulate the Wnt signaling pathway, and this mouse was instrumental for us to show that if we modify the Wnt signaling pathway in a way that we cannot modify any longer the gut vascular barrier, we also block the migration of microbes from the gut into the liver, we decided to use the same model system in order to block the interaction between the gut and the brain with what we call now the new vascular barrier, the choroid plexus vascular barrier. And this mouse, it's a mouse in which everything happens only in the endothelial cells. Beta-catenin becomes activated after treatment with tamoxifen only in endothelial cells, and we have shown that this is blocking bacteria from getting from the gut into the liver, and presumably also upper up into the brain. And so we use these mice and we analyze what is the status of the choroid plexus vascular barrier. So we gave tamoxifen, and then we analyzed 7 days later after recovery what was the expression of PV1, and we found that this PV1, which is normally in the negative control, is open, as we would expect after the decrease. So when we are activating beta-catenin, then we are also mimicking what we were seeing with inflammation, but in this case we don't have inflammation, so we are closing down the vessels just by modulating PV1 via the Wnt signaling pathway and beta-catenin activation. So now we have a perfect model system to see what happens in the brain when we have we avoid this interaction between the gut and the brain. Since we started that patients with IBD, acute IBD, have depression, we looked at whether these mice actually had depression. And so this is what we did, and we did these experiments of behavior in these mice. Sorry. And we found that the mice in which we closed down this interaction, we closed down the PBB, but also the GVB, then they have an anxiety-like behavior. They stay more in the dark chamber, and they also have an impairment in episodic memory, so they have more difficulties in distinguishing between a foreign object and a novel object. So suggesting that even in the absence of inflammation, when we block the interaction between the gut and the brain via the choroid plexus vascular barrier, we are mimicking what happens in patients with acute IBD that are undergoing an anxiety-like behavior or depression. So in conclusions from this last part, I hope I've convinced you that during intestinal inflammation, this leads to an alteration of the gut vascular barrier and the choroid plexus vascular barrier. So under normal conditions, we have an interaction between the brain and the rest of the body, and this interaction allows the brain to receive signals from the periphery. During inflammation, what happens is that the gut vascular barrier becomes compromised. So there is an increase of permeability. This allows inflammatory molecules to migrate from the gut into the liver, into the brain, but also microbial molecules to get into the brain. And the brain, in order to protect from further inflammation, closes down the choroid plexus vascular barrier. And this is the price that we have to pay to avoid inflammation to get into the brain, because this leads to depression and anxiety-like behavior. So during intestinal inflammation, we first have an increase of inflammatory molecules which get also into the brain, and then to protect the brain, the endothelial barrier is closing down. PV1 is not detectable any longer, and this correlates with the inability of the molecules to get from the periphery into the brain, and this leads to depression. So let me thank my collaborators for the first part, Alice Bertocchi, and for the last part, Sara Carloni, and this was done in collaboration with the people from Humanitas Research, Michela Matteoli and Simona Lodato, with Antonella Borreca and Sara Mancinelli. Ilaria Spadoni did all the initial identification of the vascular barrier in the gut. And I want to thank my collaborators, everybody, and you for being here to listen to my talk.
Thank you so much, Maria. I enjoyed every part of your talk. Very amazing insights on the gut-brain barrier and breaking new ground there. Thank you so much. It was very illuminating. So I I have questions, and I'm sure the audience have questions, but as always here in this forum, we ask questions via Twitter. So let me share my screen so I can remind everyone how to ask the questions via Twitter, and remind we do this via Twitter, so we allow people connecting from different time zones and watching the talks in YouTube to also ask questions in an asynchronous manner. And so for questions to Maria, please search for the account Global IMMUNOTALKS. You will find there already a tweet that says ask questions for Dr. Maria Rescigno here. Reply to that tweet with your question and mention the hashtag Global Immuno, and Maria will answer the questions using her own personal account, and yeah, take the opportunity to interact with Maria in Twitter. So thank you, Maria, again. We really appreciate your efforts for presenting your fabulous research here in this forum.
Thank you, Elena. And I actually have to really compliment on this Global IMMUNOTALKS. I think it's a great initiative. So I want to compliment you and all the others, and also Matteo Iannacone.
Thank you, Maria, our pleasure. So with that, we will close the session for today, and we hope everyone will join next week as well for the Global IMMUNOTALK by Scott Muller. Thank you so much. Bye.
Bye-bye. Bye, everybody. I will answer your questions.
Thank you, Maria.
Bye-bye.
Bye-bye.